Bismillahirrahmanirrahim. Assalamu alaikum dear students. I hope all of you will be fine. I am Dr. Muhammad Ali Hashmi from University of Education Atak campus, and we are studying the subject of Introduction to Biochemistry. This is lecture number 19, and the topic of this lecture is Chemistry and Biological Importance of Fatty Acids, Waxes and Glycerides. These three are basically storage lipids, so we are going to study the chemistry and importance of the storage lipids in this lecture. So the main concepts of this lecture would be the importance and chemistry of the following storage lipids. Just as a brief concept, what are the storage lipids? So we will be studying two different kinds of lipids. The first one are the storage lipids, and the other one will be membrane lipids. Membrane lipids would be the lipids which have a direct relation to biological membranes. They are found in the biological membranes. They have various functions in the biological membrane to they help in letting the substances pass through the membrane, and they actually guard or protect the cell contents to keep them in a compartment. So those lipids will be covered in the membrane lipids. The other one which we are going to study in this lecture, these would be the storage lipids. Storage lipids, basically, as the name implies, they actually store energy. They are there to store energy in their structures, and we are going to study in the next few slides how do they do that and how it relates to their structure. So as we studied in the previous lecture about fatty acids, how they are formed and what is their basic structure, in this lecture we are going to describe its chemistry a bit more, the nomenclature of fatty acids, how do we name these compounds, and then how do they have more energy than the carbohydrate counterparts. And then we are going also to describe their other types, that is waxes and glycerides. So as a fact, the fats and oils used almost universally as stored forms of energy in living organisms are derivatives of fatty acids. You know that the fats and oils, they are universally means that in almost all living organisms, these fats and oils, their function is to store energy. They are the stored forms of energy. And what are these fats and oils? They are derivatives of fatty acids. So fatty acids would be their main component. Whenever you hear of a fat or an oil, you will see that the fatty acid is the main component of that proper fat or oil. These fatty acids are hydrocarbon derivatives at about the same low oxidation state, that is as highly reduced as the hydrocarbon in fossil fuels. So that is the main thing. That is the main feature of fatty acids which distinguishes them, or which makes them superior energy storage materials compared to the carbohydrate counterparts. So what does that sentence actually mean? These the fatty acids are about the same low oxidation state as in hydrocarbon fossil fuels. As you might have seen in the basic organic chemistry which you have studied in the FSC and intermediate classes, that what are the structure of fossil fuels? For example, gasoline. You have seen that is also a hydrocarbon chain. It's a branched chain of hydrocarbons. So it is highly reduced, mean it has a lot of hydrogens in it and it doesn't have many oxygens. So upon oxidation, it can release a lot of energy, which we can use in different forms, like to run the engines in our vehicles and other purposes. Similarly, if you look at the glucose, for example, that is a carbohydrate, it has a lot of hydroxyl groups in it. So it is already greatly oxidized. It has energy which it can give on further oxidation, but the amount of energy given by that compound and a fatty acid would be quite different, because fatty acid is at a very low oxidation state, so it can be oxidized and can give many folds the energy compared to a carbohydrate counterpart. So this cellular oxidation of fatty acids, the oxidation which happens in the mitochondria, that is called cellular oxidation. So when fatty acids get oxidized in the mitochondria, the products are carbon dioxide and water, and it is the similar as, you know, when in a combustion engine the fossil fuel burns, what is the product? The product is carbon dioxide and water. So it looks as a similar process, which gives you energy and releases carbon dioxide and water. So fatty acids behave the same way as the fossil fuels behave in an engine. That is why they are preferred stored forms of energy in a living organism. So we are going to discuss the chemistry and biological importance of the fatty acids most commonly found in living organisms. So two types of fatty acid containing compounds, the triacyl glycerols and waxes. These will be described with the focus on their chemistry and biological importance in this lecture. So we come to the fatty acids. Fatty acids are the carboxylic acids, as we have discussed in the previous lecture, that they have a long hydrocarbon chain and a carboxylic group at the end. So they're basically carboxylic acids, and their hydrocarbon chain, it ranges from 4 to 36 carbon atoms. That is a common range in which they are found, and so their formula will be definitely C4 to C36. It will be somewhere between these numbers. In some fatty acids, this chain is unbranched, mean it is straight chain and fully saturated. What does that mean? That it has no double bonds in it. All the bonds are single bonds in some fatty acids, so they are called fully saturated, and they are unbranched, mean they are straight chain compounds. And in some other fatty acids, the chain may contain one or more double bonds. So double bonds are also found in some fatty acids. And a few contain 3 carbon rings, hydroxyl groups or methyl group branches. Some fatty acids, like those ones found in bacteria, they might contain different other groups on them, like these ones, the hydroxyl group, 3 carbon rings and other methyl group branches. And fatty acid, they are called storage lipids. So that is a specific name for them. The storage lipids, fatty acids are known as storage lipids because, as we discussed on the previous slide, they have a lot of energy stored in them, and upon oxidation they release that energy. That is why they are called storage lipids. Now we move to the nomenclature of fatty acids. How do we name these compounds? In the previous lecture we have studied this topic very briefly, so we are going in a bit more detail here. The nomenclature is actually very simplified for these compounds. It specifies the chain length and number of double bonds separated by a colon. For example, a 16 carbon saturated acid, that is palmitic acid, how it will be abbreviated. It is a fully saturated carboxylic acid. It means there are no double bonds in it, and it has 16 carbons long chain. So it will be abbreviated as 16:0. 16 mean the chain length is 16 carbons, and the colon 0, that means that there are no double bonds in this compound. And the 18 carbon oleic acid with one double bond, it will be represented as 18:1. So now when we know that there is a double bond, where is that double bond? There must be its position, and it should be specified so we know at which carbon atom the double bond exists. For the positions of these double bonds, we specify them by superscript numbers following delta. So this symbol delta is used with a superscript on it, which tells us the position of the double bond. Let's say a 20 carbon fatty acid with one double bond between carbon number 9 and carbon number 10. So how do we number these carbon 9 and 10? We know that the numbering starts from carbon number 1, which is the carboxylic group. So the double bond is between carbon number 9 and 10, and the other double bond in it, so this compound has two double bonds. The first one is between carbon number 9 and 10, and the other one is between carbon number 12 and 13. How will we represent that? Like this: 20:2. 20 means it has 20 carbons in it, and 2 after the colon means there are two double bonds. And then in the parentheses, delta and superscript 9:12. That means that the positions of double bonds are at carbon number 9 and at carbon number 12. So that is how we represent these fatty acids, and their nomenclature works. So here is a table showing some naturally occurring fatty acids, their structural properties and nomenclature. We're not going to spend a lot of time on this. You may actually have a look at all the acids and their common names and different properties like melting and boiling points and solubility in water. For example, if you look at myristic acid, its systematic name is N-tetradecanoic acid. That is the IUPAC name. Here's the structure, and 14:0 mean it has 14 carbons in it. There is no double bond. If we move to this one, oleic acid, its systematic name would be cis-9-octadecanoic acid, because it has double bond, and its skeleton would be 18:1, and delta 9 means the double bond is at the 9th carbon atom. And similarly we can move on. In arachidonic acid, you see that 1, 2, 3, 4, there are 4 double bonds in the arachidonic acid. So there is a common pattern in the location of double bonds in these fatty acids, and in most monounsaturated fatty acids the double bond is between carbon number 9 and 10, and the other double bonds of polyunsaturated fatty acids are generally delta 12 and 15. So this pattern is common in the monounsaturated fatty acids. What does that mean? Monounsaturated mean with mono mean single, unsaturated which have a double bond. So that means the fatty acid which have a single double bond, that mean one double bond, which have one double bond in them, they will be called as monounsaturated fatty acids. And mostly in monounsaturated fatty acid, it is seen that the double bond is between carbon number 9 and 10. And if we have polyunsaturated fatty acids, then there are definitely more than one double bonds, and the other double bonds locations are generally, this is generally, it is not always the case, but mostly these other double bonds are between carbon number 12 and 13 and 15 to 16. So we have an exception to this, that is arachidonic acid, where the double bonds are at 5, 8, 11 and 14th carbon. So that is an exception to the this generalization we just made. The double bonds of polyunsaturated fatty acids they are almost never conjugated. Conjugated mean that one double bond and a single bond, and then again the double bond comes, like this one. This is a conjugated double bond. So in fatty acids, the double bonds are not conjugated commonly. They are like this. They are far apart from each other. And in nearly all naturally occurring unsaturated fatty acids, the double bonds are in the cis configuration. So this is another generalization, that it is found that in almost all the fatty acids that occur naturally, the double bonds are cis. They are not trans in their configuration. And trans fatty acids are produced by fermentation in the rumen of dairy animals and are obtained from dairy products and meat. The fatty acids which have trans double bonds in them, they are called trans fatty acids. Where are they produced? They are produced in the rumen. Rumen is the first stomach. You know that dairy animals like cows and buffaloes, these animals have 4 chambers in their stomach, or you may call 4 stomach parts. So in the first stomach, that is called the rumen. In that one, due to the fermentation process, trans fatty acids are produced. So that is why they might be present in the dairy products and meat of these animals. Trans fatty acids are also produced during hydrogenation of fish or vegetable oils. When we when we heat up the vegetable oils and fry the fish in them, or even heating the vegetable oil again and again, that may produce trans fatty acids in it. And you know that diets high in trans fatty acids, they correlate with increased blood levels of LDL, that is a bad cholesterol, and decreased HDL. HDL is the good cholesterol, and it is recommended that one avoids large amounts of these fatty acids. What is LDL? It is the low density lipoprotein, and HDL is the high density lipoprotein. Low density lipoproteins are not good for our health, and they actually make the arteries clogged with the cholesterol. So we should avoid diets which are high in the trans fatty acids, and we should take the diets which are with the high density lipoprotein, and this HDL is a good cholesterol for our health. And unfortunately, you see these things, French fries, donuts and cookies tend to be high in trans fatty acid. So I have a bad news for you. For French fries lovers, donuts lovers, and cookies, when you fry these things in vegetable oils, many cis fatty acids can change to trans fatty acids. So better to avoid these foods for your health, and even if you like them, you can have them very few times, but not too often. So health comes first, and I believe that you guys would be avoiding these foods from today after this lecture. Now we come to the solubility of fatty acids. You know that fatty acids and their properties are largely determined by the chain length, carbon chain length, and the degree of unsaturation which is present in that hydrocarbon chain. So these are two main factors which determine the solubility or other properties of the fatty acids. The number one is their carbon chain length, and then the degree of unsaturation, how many double bonds are present in them. They are the main factors which determine their properties. The nonpolar hydrocarbon chain, the chain which is apart from the carboxylic part, that accounts for the poor solubility of fatty acids in water. As you know that the hydrocarbons, they are organic compounds, they are insoluble in water. So if you look at the fatty acid structures, it has a carboxylic group and a long chain of hydrocarbon. So that hydrocarbon chain is mainly, that is insoluble in water, and that is found in more amount. मीन्स ज़्यादा जो उसके अंदर है वो हाइड्रोकार्बन चेन है और कार्बोक्सिलिक एसिड पार्ट थोड़ा सा है. तो इट मीन्स कि जो ज़्यादा वाला पोर्शन है वो ज़्यादा कंट्रीब्यूट करेगा, एंड दे विल बी insoluble in water due to their hydrocarbon chain. The carboxylic part is the one which makes it soluble in water because it can ionize and it has some interaction. So it will be very sparingly soluble, poorly soluble, because carboxylic acid part is very little and the hydrocarbon chain part is the major one. If you look at, for example, lauric acid, it has 12 carbon in it and its molecular mass is 200. It has a solubility in water of 0.063 milligram per gram of water. That is very, very poor solubility. But if you compare it to that of glucose, glucose has also a molecular mass of 180, that is around similar to the lauric acid, and it has solubility in water 1100 milligrams per gram of water. Did you see the difference? So that is because glucose is highly oxidized. It has a lot of OH groups which can interact with water and it can get dissolved in water, while the lauric acid, it has a 12 carbon long chain in which only one carboxylic group at one end. The other hydrocarbon chain makes it insoluble or very poorly soluble in water. The longer the fatty acid acyl chain and the fewer the double bonds, the lower is the solubility in water. That is the rule. You remember it. जितनी लंबी fatty acid chain होगी, जितने कम double bond होंगे, compound water में उतना ही कम soluble होगा. Carboxylic acid group is actually polar and ionized at neutral pH, and that is the one which accounts for the slight solubility of shorter chain fatty acids in water, as we discussed already. Next, we move on to the melting points of fatty acids. Fatty acids are strongly influenced by the length, their melting points are influenced by the length and degree of unsaturation of the hydrocarbon chain. At room temperature, that is 25 degrees Celsius, the saturated fatty acids from 12 to 24 carbon atoms have a waxy consistency. They appear like a wax, a very thick liquid. Whereas unsaturated fatty acids of these lengths are oily liquids. And if you take the same length fatty acids which have unsaturation or double bonds in them, they appear as oily liquids, like the oils we used at homes, soybean oil and other vegetable oils. You have seen that they appear as liquids. They do not appear as solids. So what does that mean? That means that they have double bonds in them. And butter and other such things which appear as solids, they are the saturated fatty acids. And why is this difference in the melting points? The unsaturated ones are already melted and the other ones are solid. That is because of different degrees of packing of fatty acid molecules. We see on the next slides what does that packing mean. In the fully saturated compounds, the free rotation around each carbon atom bond gives the hydrocarbon chain great flexibility as we have discussed in the previous lecture about the conformational flexibility and different conformations. You know that the saturated compounds have great flexibility. And the most stable conformation is the fully extended form. Fully straight chain form of these is the stable conformation, because it gives it less steric hindrance and steric interactions of the neighboring groups. And due to that ability, these saturated compounds can pack together tightly in nearly crystalline arrays with all atoms all along their lengths in van der Waals contact with the atoms of neighboring molecules. Here you may see that this long chain hydrocarbon that prefers to live in this condition, and due to this long chain, straight chain condition, they can pack like this. You see on the right side, saturated fatty acids can pack in closed arrays, and due to this close packing, there will be van der Waals interaction between these carbon atoms of the neighboring carbon chains, and due to this close packing, they appear to be solid. You know that the solids have close packing of atoms in them, and liquids do not have close packing. Okay, so saturated fatty acids, they have a close packing of these carbon chains that makes them appear solid, or that makes their melting points higher. If you look at the unsaturated fatty acids, a cis double bond, if there is a double bond present, a cis double bond, that makes a kink. That means a sharp twist in the hydrocarbon chain. Fatty acids with one or more several such kinks cannot pack together as tightly as fully saturated fatty acids, and their interactions with each other are therefore weaker. So that means that if there is a double bond in the chain, that bends the chain, and then it is difficult for these carbon chains to pack together, as we have seen previously that saturated carbon chains, they can pack together much easily and tightly. So when they cannot pack very well together, it takes less thermal energy to disorder these poorly ordered arrays of unsaturated fatty acids. So they have markedly lower melting points than saturated fatty acids of the same chain length. Have a look here. This is a chain with one double bond. Have you seen that it should have gone like this here, but due to the double bond there is a kink, and this bend has made this hydrocarbon chain a bit in this bend form, and when they are mixed, you see how loose is their packing. They cannot pack as tightly as we have seen in the saturated carbon chains. How can they pack tightly? So due to this loose packing, the thermal energy that is required to break these apart, that is very less, and their melting point will be less. They will be easily disordered as compared to the saturated fatty acids. That is why their melting points are not very high, and they are low. We look at the some food fats. Take for example olive oil, butter, and the beef fat. If we compare them, they contain mixtures of triacylglycerols differing in their fatty acid composition. So definitely there are three different kind of fats, and their fatty acid composition is different. If you look at the melting point of these fats and their physical states at room temperature as a direct function of their fatty acid composition, we have these three conclusions. Olive oil has a high proportion of long chain, that is carbon 16 to 18 unsaturated fatty acids, which accounts for its liquid state at 25 degrees centigrade. You know that olive oil is a liquid oil, so that is due to its unsaturated fatty acids. And the higher proportion of long chain saturated fatty acids in butter increases its melting point. So the butter is a soft solid at room temperature. Butter has a high proportion of long chain saturated fatty acid, which increases its melting point due to close packing of those carbon chains, and it appears as a solid at room temperature. And lastly, if you look at beef fat, that has an even higher proportion of long chain saturated fatty acid. It is a hard solid. Compared to butter, if you look, you can cut butter easily with a knife, but if you compare beef fat, that is not as easy as cutting butter with a knife. That is due to higher proportion of long chain saturated fatty acids, which make it a hard solid. Next, we see the oxidation of fatty acids. When lipid-rich foods are exposed too long to the oxygen in air, they may spoil and become rancid. It means that if the foods which contain lipids in them, you keep them outside in the air for a long time, they will develop a smell and they become rotten. The unpleasant taste and smell associated with that rancidity, it is the result of oxidative cleavage of the double bonds in unsaturated fatty acids, which produce aldehydes and carboxylic acids of shorter chain length and therefore high volatility. So you know that these aldehydes have different smells, and the bad smell in that rancid fatty acid would be due to the production of these aldehydes in it, which are highly volatile, and they are the reason of that bad smell. And let's have a look at the fatty acids in 3 dimensions. Here you can see the models of palmitic acid, oleic acid, and linoleic acid. Palmitic acid is a saturated 16 carbon fatty acid, and you see that it has a straight long carbon chain which is shown here, and due to which it can pack easily and strongly like a saturated fatty acid. And if you look at oleic acid, it is 18 carbon fatty acid, and it has a cis double bond here which makes its structure bent like a V shape. Have a look and differentiate between palmitic and oleic acid, the difference in their structure. And then we look at linoleic acid. It has more than 1 double bond. It has 2 double bonds in it, and both of them have cis configuration. And look how its structure becomes even different from the oleic acid, which is its counterpart. They both have 18 carbons, but oleic acid has 1 double bond and linoleic acid has 2 double bonds in it. So that creates a difference in their structure in 3 dimensions. Next topic is waxes. What are waxes? Biological waxes are esters of long chain carbon 14 to carbon 36 saturated and unsaturated fatty acids with long chain alcohols. When these long chain alcohols they react with the long chain fatty acids, they produce esters. We have studied in the previous lecture that combination of alcohols and carboxylic acids produce esters. This is an ester. So this portion palmitic acid, this portion came from palmitic acid, and this was 1-triacontanol. That is an alcohol. So when they condense together after producing a water molecule, this group C double bond O, then single bond O, and then single bond hydrocarbon chain, that is the ester functional group. So waxes are basically these esters. You can see an ester here on the right hand side, which is rotating in front of your eyes. So melting point of waxes range between 60 to 100 degrees centigrade. They are generally higher than those of triacyl glycerols. In plankton, what are plankton? The diverse collection of organisms that live in large bodies of water and are unable to swim against a current. Yeah, these are the organisms which live in oceans, and they don't swim against water, so they are mostly staying at a place. The free-floating microorganism at bottom of the food chain for marine animals. In these organisms, waxes are the chief storage form of metabolic fuel. So these planktons and the microorganisms which are at the bottom of food chain for marine animals, they have waxes as a storage of energy in them. So waxes also serve a diversity of other functions related to their water repellent properties. Wax is basically water repellent. You might have seen different kind of waxes in the market which are used for different purposes. We'll see an example in the next slides which show you their water repellent properties. Certain skin glands of vertebrates secrete waxes to protect the hair and skin and keep it pliable, lubricated and waterproof. So there are different vertebrates whose skin glands produce these kind of waxes to protect the skin, hair and keep it waterproof and lubricated. Birds, particularly waterfowl, you see the strong swimming birds, for example ducks, they have these kind of glands in their skin which secrete waxes through their preen glands and keep their feathers water repellent. So you have seen that ducks, they float on water, but still they don't get wet. Like if you put a hen in the water, it will really get wet. Its feathers will really get wet, and it won't be able to fly or swim anymore. But if you look at the ducks, they have these special wax glands that makes their feathers water repellent. So when they come out of water, they just throw the water off their feathers very easily, and they look dry, they don't look wet. So similarly in some plants, the shiny leaves, for example shiny leaves of holly, rhododendrons and poison ivy, these are different plants whose leaves appear to be very shiny. They are coated with a thick layer of waxes which prevents excessive evaporation of water and protects against parasites. So these plants, have a look at the leaves of holly, how shiny they are. They are due to the thick layer of waxes on them which is produced by the plant. These waxes protect the extra water loss due to evaporation and transpiration, and also protect them from the parasites. So parasites would be repelled by the smell of these waxes on these leaves. Similarly, you look at the poison ivy plant and the rhododendrons, their leaves are quite shiny due to the presence of these waxes. And biological waxes, they find a variety of applications in the pharmaceutical, cosmetic and other industries. For example, lanolin, that is from lamb's wool, beeswax, carnauba wax, that is from a Brazilian palm tree, and the wax extracted from spermaceti oil from whales. These are widely used in the manufacture of lotions, ointments and polishes. I have given a reading slide at the end. That is the third last slide. It is for you to read, and it has an interesting information in it about the spermaceti oil and these whales. So have a look and read it. Next is the glycerides, the last storage lipid we are going to discuss in this lecture. Glycerides are more correctly known as acyl glycerols. These are esters formed when the glycerol and fatty acids they react together, and they are very hydrophobic. You see that glycerol has 3 hydroxyl groups in it, which can be esterified with 1, 2 or 3 fatty acids to form mono, di and triacyl glycerols. If one OH is acylated, it will be a monoacyl glycerol. If 2 of them are acylated, it will be a diacyl glycerol, and if 3 of them, then triacyl glycerol. The simplest lipids constructed from fatty acids are the triacyl glycerols, also referred to as triglyceride fats or neutral fats. You see an example of triacyl glycerol here. This is triacetin, and that is the simplest possible fat after triformin. It has a very small fatty acid ester of the glycerol molecule. So this is triacetin. The glycerides containing the same kind of fatty acid in all 3 positions are called simple triacyl glycerols and are named after the fatty acid they contain. Simple triacyl glycerols of 16, 18 and 18:1, for example, are triasterine, tripalmitin and triolene respectively. So we have an example of tripalmitin here that has 18 carbons in it. You see this is the triacyl glycerol, where the acyl group comes from the palmitic acid. So all 3 OHs are acylated by the palmitic acid. That is why it is called as tripalmitin, and it is a simple glyceride. Most naturally occurring triacyl glycerols are mixed. They are not simple. They contain 2 or more different fatty acids. For example, have a look at the example here. These compounds are named according to the position of each fatty acid. If you look at this, this is the basic structure of the glycerol. So carbon number 1, 2 and 3. So at carbon number 1, we have a stearic acid moiety, which is esterified with the help of this OH group. On carbon number 2, we have moiety from linoleic acid, and on carbon number 3, the acyl group comes from the palmitoleic acid. So its name would be 1 stearoyl, 2 linolyl, 3 palmitoyl glycerol. So that is a mixed triacyl glycerol, which is named according to the different fatty acids from which it has generated. As the polar hydroxyls of glycerol and the polar carboxylates of the fatty acids are bound in ester linkages, the triacyl glycerols are non-polar hydrophobic molecules which are essentially insoluble in water. You know that the polar groups of glycerol, that is OH group, and the carboxylic acid OH group, they have condensed together and released a water molecule. So now this is a non-polar molecule, which the glycerides would be insoluble in water due to this non-polarity. And lipids have low specific gravities than water, which explains why mixtures of oil and water, for example, oil and vinegar salad dressing, they have 2 different phases. Oil with the lower specific gravity floats on the aqueous phase. So if you mix the oil and water, the oil will float on the water due to its low specific gravity. Now we see the biological importance of the glycerides. In most eukaryotic cells, triacyl glycerols form a separate phase of microscopic oily droplets in the aqueous cytosol. These droplets serve as depots of metabolic fuel. So many eukaryotic cells have triacyl glycerols. They are floating in the cytoplasm and they have stored energy in them. In vertebrates, specialized cells called adipocytes or fat cells store large amounts of triacyl glycerols. uh, or as fat droplets that nearly fill the cell. If you look at this example, here is structure of an adipocyte shown, where you can see that this is a big droplet of triacylglycerol which has filled the cell entirely, with its other organelles squeezed to one side. Triacylglycerols are also stored as oils in the seeds of many types of plants, providing energy and biosynthetic precursors during seed germination. So many seeds have triglycerides in them, which when germinate, they use the energy of these triacylglycerols. Have a look, for example, here. Here is the cross section of a seed cell, where you can see droplets of triacylglycerol stored in it. And these cells then have the enzymes called lipases, which catalyze the hydrolysis of these stored triacylglycerols and release the fatty acids for export to the sites where they are required for the energy needs. There are two significant advantages of using glycerides. Triacylglycerols as stored fuels rather than polysaccharides like glycogen and starch. The first one is because the carbon atom of fatty acids are more reduced than those of sugars, which we already discussed in the start of this lecture. The oxidation of triacylglycerol will yield more than twice as much energy as the oxidation of carbohydrates. And the second advantage is because triacylglycerols are hydrophobic and therefore unhydrated, and they don't have to save the water of crystallization or water of hydration with them, so they can be stored without carrying extra weight of water. In humans, humans have fat tissue that is composed primarily of adipocytes under the skin and in the abdominal cavity and in the mammary glands. So humans have a lot of fats in their bodies, and moderately obese people, मतलब जो बहुत ज़्यादा मोटे ना हो, बिल्कुल normal से मोटापे वाले people, they have 15 to 20 kg of triacylglycerols deposited in their adipocytes. They could meet their energy needs for months by drawing on their fat stores. तो अगर ये महीनों तक चाहें तो इनके जो अंदर फैट स्टोर्ड हैं उनसे इनको एनर्जी मिल सकती है. And in contrast, if you look, human body can only store less than a day's energy supply in the form of glycogen. Carbohydrates such as glucose and glycogen do offer certain advantages as quick sources of metabolic energy, one of which is their ready solubility in water. So difference क्या है कि जो carbohydrates हैं और glycogen वगैरह है, वो उनका advantage क्या है, वो body क्यों store करती है उनको, कि वो quick source of energy हैं, वो फौरन water soluble हो जाते हैं और quickly आपको energy देते हैं. Fats जो हैं वो आपको इतने quickly energy नहीं दे सकते. That is why fats are stored and carbohydrates are used mostly. So we have to eat food more than once a day to recover the supply of carbohydrates and glycogen in our body. In some animals, triacylglycerols stored under the skin, they serve not only as energy store but as insulation against low temperatures. Like if you look at seals, walruses, penguins and other warm-blooded animals, they are abundantly padded with triacylglycerol. Their skins have, under their outer skin, they have thick layers of triacylglycerols, which also have energy stored in them, and they also work as insulators and protect them from the outside cold environment. In hibernating animals, for example bears, the huge fat reserves accumulated before hibernation, they serve the dual purpose. They serve the purpose of insulation, and they also serve the purpose of energy for the animal. So here is the slide I told you earlier that is for your information. You have to study it and see that what is the significance of these triacylglycerols in these sperm whales and what functions they perform, how much it is stored in them. This is for your information. You have to study it yourselves. And at last, you are advised to visit the following link. It has many beautiful 3D models, as I have shown you some of them in the previous slides. So these models can help you understand how the shapes and the packings of these 3D materials can happen together. So this website will give you very useful three-dimensional models of these compounds. And here are the references. These are the books which were consulted for the preparation of the lectures. I hope you have got and understood the major points of this lecture. If you have any questions, you are free to ask them in the Google Classroom section, comment section. And we're going to study about the membrane-bound lipids in the next lecture. Until then, Allah Hafiz, fi amanillah.